Hello, everyone. I am Dan Krotz with Berkeley Lab Public Affairs, and thanks for attending our third installment of Sit Down with Sabin, a weekly conversation in which Sabin Russell, sitting right here, engages lab staff about the ins and outs and ups and downs of innovative science. Today, you'll hear about the carbon cycle. Of course, we also want to hear from you, so there'll be plenty of time throughout the talk for questions. And now, before we begin, a little bit about our moderator, Sabin Russell. He joined Berkeley Lab just last year after a career as a Bay Area health and science reporter. This included 22 years with the San Francisco Chronicle, freelance work for the New York Times, and a Knight Science Journalism Fellowship at MIT. He's best known for his work covering HIV both in Africa and the US, as well as the tsunami in Sri Lanka and the Columbia shuttle disaster. He is now at CSO, Creative Services Office, as one of their lead writers, where you can use him to help you write your documents as well as develop red products and things like that. So he's up for hire now here at the lab. And without any further ado, the stage is now yours, Sabin. Thanks.
Thank you, Dan. Today, I think, is almost exactly my first anniversary at the lab as a writer and editor. And I still have the same feeling I had in the first weeks I was here, that this lab is an extraordinarily impressive place, in part because of the diversity of the science that goes on here. We have the nanotechnologists at the Molecular Foundry, and we have the cosmologists just down the hall. We pursue a lot of high-energy physics here, and we also have people who are perfecting the low-energy building. I think that's one of the great strengths of this place, but this particular afternoon experiment that we're doing is an attempt to sort of broaden the message to all of the labs so that people who may be working very diligently in one aspect of the science can get some exposure to the work that's going on in other places. We also have a very diverse group of people here in the summer. We have student interns, we also have postdocs, and we have staff scientists. So this is a conversation format, again, a bit of an experiment, but the idea is maybe we're going to dial down the science just a bit and talk in more general terms about what we're doing to people like all of us who have an interest in science. And it's simply a way to celebrate the technology of this lab, the new and interesting and important things that are done here. And so our topic today is going to be the carbon cycle. And we are extremely lucky to have a person who is described as a national expert on terrestrial ecosystem, on how terrestrial ecosystems affect the carbon cycle. Margaret Torn is a local girl. She grew up in Marin County, attended the College of Marin, transferred to Cal, graduated from Cal, and then did her PhD at Cal in energy and resources. Still have to use my notes. And she joined the lab in 1998 after postdocs at Stanford and UC Irvine. And she's now a staff scientist with the Earth Science Division. And she's program head for climate and carbon sciences. So I'd like you all to give a big hand to Dr. Margaret Torn. So I thought, Margaret, that since we have a broad audience here, I think most of us know what the carbon cycle is, but very briefly, explain to us what the carbon cycle is in terms of your research and how it's relevant to climate studies today.
Let's see, since it's lunchtime, we can start with the carbon cycle that everyone here is experiencing now, which is the productivity of terrestrial ecosystems producing the food that we eat. The other way that we always experience the carbon cycle is through the amount of CO2 that's in the atmosphere, which is making it a bit warm today. But as how we study it, the carbon cycle is the study of the element carbon as it is cycling from being inorganic CO2 in the atmosphere to being photosynthesized by plants. The plants die or shed organic material that goes into soil. Microbes decompose that. respiring, turning the CO2 back to the atmosphere, so you have a cycle. And the same thing is happening in the ocean. So we're studying that, and we call that a biogeochemical cycle because it's involving biological process, physical process, chemical and geological processes. So that's
when we talk about scientific research of any kind, people often use sort of the metaphor of the journey of discovery. And one of the things that I think is most interesting about you is that you are literally traveling to do your work. Margaret Torn is a field researcher. She's not stuck at the bench like many people here, and runs up a lot of frequent flyer miles and probably has built a terrible carbon footprint as a result of that. But your work has taken you, as I understand it, to the North Slope of Alaska, to Hawaii, tough job.
Exactly.
Russia, to the Swiss Alps, to the Colorado Rockies. So why is it that you go so many places to do this work?
Oh, that's a good question. So the reason why we go might be surprising in a way, I guess, that you might think, well, we go because we need to study each place, but really we're mainly interested in the generalizable theory of how does the carbon cycle work? What controls these processes? And in any soil, anywhere in the world, you have microbes doing their work, and they're decomposing organic matter and transforming it. That releases CO2, which is well mixed, and so it's a very local phenomena. You have to go somewhere to study it, but it's a global phenomena that you've got this global CO2. And we go where you can study it. It's a really complex system, and you have to be very opportunistic and look for those natural experiments where you can go and actually disentangle the different factors that are affecting ecosystems. And that's the main reason, but there are some hot spots.
So the common denominator of most of these places where you do your work is soils.
Yeah.
And I think a lot of times when people think of the carbon cycle, they think of water, they think of the ocean. Mm-hmm. How important a role do soils play in the carbon cycle, which, spoiler alert, I've been told is one of the least understood of the aspects of the carbon cycle.
So we have all this sort of hidden carbon cycle that's down in the soil, but the soils hold more than twice as much carbon as there is in the atmosphere.
Twice as much? Twice
as much as there is a CO2 in the atmosphere. And it's very dynamic. Microbes can be decomposing it, releasing it as CO2. You have new carbon coming in from plants, so it's active and dynamic, but it's really hard to study because it's kind of hidden. It's no easy way to see what's happening. So we try to choose hot spots. You go up to the North Slope of Alaska because there's so much permafrost carbon. That's an interesting case. Or you go to the Alpine because that's going to change a lot, and you want to see if trees will be marching uphill into this new, warmer alpine environment in the future. So
let's talk about some specific places that you've been. One of the things that intrigued me was the work that you did in Russia, and that had to do with some archive samples from a Russian scientist of 60, 80 years ago. Yeah. Tell us about that.
Okay, so this is a bit of a history, but in 1997, I went to Russia, went on a field trip driving from St. Petersburg to Voronezh with an ex-KGB officer at the wheel and some other Russian soil scientists along, and we dug soil pits. But the history is that 100 years before then, really wonderful soil scientist and ecologist, Dr. Rizbo Lazinski, had done the same tour and saved these perfect soil monoliths, which were then archived in St. Petersburg. And just after the fall of the Soviet Union, we had access to them.
How large were these monoliths?
So they were about as tall as this backdrop, 6 feet, about this wide
sort of
across.
Rectangular core samples?
Yeah, so they took not a core, it's like a perfect slab with a plastic front so you could look at it, or glass, and you could look at at that time it was glass. Look at the soil profile.
And what happened to his work?
So he brought his soil collection to one of the first or second World's Fair in Paris just before 1900. He won a gold prize for natural history work, but he was politically out of favor with both the Bolsheviks and after the revolution, and he was actually executed, and his work was never published. So his logs were there in the Academy of Hydrology in St. Petersburg.
But the work was preserved and the samples were preserved.
exactly.
So what sort of things did you find out in your research, and does it have a bearing on the work you would do here?
Well, obviously this was a unique opportunity to actually see how soils had changed. So we knew where they were collected. We went to the exact same spot, collected soil, and compared them to this 100-year-old soil. And as a general thing, the neatest thing was just learning how soils record history. That after 100 years, the trace metal work on weapons in the area was then recorded in that soil. We could see what metals they had used.
These are nuclear weapon tests?
No, these are more the building of weapons that use trace metals.
Oh, I see.
Just looking at what had happened in the area. You can look at changes in rainfall. And in our case, we looked at the carbon cycling as well. We did all those tests, but I did the carbon cycling. And the main thing that we found that has bearing now is that black carbon, the pyrogenic carbon, something we call biochar, comes from forest fires, has been considered inert. But we could tell in this system, which had been 100 years without fire, that it decomposes quite well. So that leads us.
So the carbon actually breaks up and is taken in by the microbes, or?
Yeah. So you could find out that just about anything a plant can produce, whether it's charred or not, there's a microbe there in the soil that can decompose it. And I don't know if people have heard of biochar, and if you think about this as a strategy for sequestration, it's being promoted, and it may have great benefits to put in pyrogenic carbon or biochar into soil. But the notion that it is inert or doesn't decompose, I think, is just wrong. And here was a place we could really test that, because we had the soil 100 years ago and we had it today.
Did you notice any really distinct differences between what you had archived and the soil? I gather you took samples in some of the same spots where you found the archived material.
Yeah, I mean, I think we were within just a few meters of where those original samples were taken. It was very careful reconstruction. And it was amazing how little change there was wherever we sampled in an environment that was still under the same vegetation cover and the same land use. You had just the exact same profile, same color strata, same thickness. And if you went to a place that had been under land use or near an area where there had been erosion, then there were big changes. And then across the whole Great Plains, the steppe of Russia, there was a change in precipitation, and you could see slight differences recorded that way, but really the same.
Were there any lessons to draw about climate change just from those samples and that 100-year picture?
Yeah, I think we're still working on that, so I would say I'll wait. But I think that you can see changes in long-term trends in precipitation reflected in the soil.
You mentioned several times microbes and microbial communities, and it seems like across many disciplines we're hearing more and more about the ecology of microbial communities, even in the guts of people. And I understand some of that research is driven by genomics, that we're now able to identify more bacteria because you don't have to isolate them and grow them. You can simply look at their genes. Are you doing any of that sort of work, or with JGI?
Yeah, so we partner with people here like Owen Brody, I think you might be here, Janet Jansen, who are really good microbial ecologists. And they're finding out that when we look at a soil, there's let's see, well this is one of my props. This is a fungus from the cafeteria. But it is a fungus nonetheless. And if you take a
field researcher,
a bit of soil, say half the size of this mushroom, in that will be about 1 billion microbes. Most of them can't be cultured. We don't know. So 1 gram of soil, if you're keeping track.
1 billion.
About 1 billion microbes, 10 to the 9th. Most of them can't be cultured, so we didn't know, well, who's doing all this? How will the microbes in soil, for example, respond to climate change? If we have warmer, wetter conditions, what will happen? If you don't even know who they are, what their physiological constraints are, you can't answer that question. So it's been really important to team up with the people who work at JGI and help us unravel that work.
So let's sort of spin the globe a bit to some other places that you've been. You mentioned the North Slope. North Slope of Alaska. I imagine that's a little different from the steppes of the former Soviet Union. What were you doing there?
And the challenges there are quite different as well. So in the North Slope, we're studying permafrost because you have so much carbon that's frozen in the permafrost, and methane release. So when you have decomposition without oxygen in any wet environment, tundra or wetlands, then that material you produce methane instead of CO2. And we're fortunate that, and that methane, I should add, for those who don't already know, is a very potent greenhouse gas, something on the order of 25 times as potent per molecule as CO2. So we care about how ecosystems release methane, how much, and what will happen in the future. So that's what we were studying in Alaska, was the methane release.
What sort of findings did you, or were there any surprises from that?
Well, I guess one thing we documented that wasn't a total surprise but hadn't been as well known was that the microbes in soil actually do us a big favor there. Although methane is produced, probably 80% of it gets consumed by microbes before it gets out of the soil. And there, at least, the primary way it gets out of the soil is through plants. Plants have these, that live in the tundra, the wetland plants, have a hollow stem inside called an parenchyma they use to bring oxygen to their roots. And sort of inadvertently, then, it's a highway for methane to get out. So actually, I brought one other prop of our work. I've been invited to.
Important tool.
This is another tool. This looks like a party straw, and it is a party straw. And when you're doing field work, you don't always know what you're going to be looking up and what you'll need. So I was on the North Slope of Alaska and wanted to understand whether this transport of methane by plants was passive or active. Did the plant have to be photosynthesizing? What was it? So we could put plants in the dark and they weren't photosynthesizing, but we also had a little tiny dining hall, little mesh hall at our field station, which was about 3 hours south of the Arctic Ocean on a dirt road. And we had party straws, so I could take the plants, clip the plants, put in the party straws, and see whether methane came out. And in fact, the rates were at least as high as with the plants. So we published this, and I think this was like I should have gone and looked it up. This was a plastic conduit, a biological conduit. I don't think I called it a party straw in the article. But in fact, it was red and white striped. This was our cafeteria, so yeah. So
did any polar bears come across these party straws sticking in the middle of the tundra?
We're planning a new trip to Alaska in a month, and Christina Castagno is there, works in my lab and does also lots of field work. And we're going through the challenges. So there, it's quite different. The mosquitoes you've heard are bad, but as we hear, they're much worse than that. The little black flies kind of line your eyes. You can have a storm coming through. You bring bear spray.
Bear spray.
It's easy to get lost because the sun isn't going east to west. It's just kind of going in a circle around the horizon, and it's a rather flat horizon. And it's not getting dark, so you're working 20 hours a day because it's expensive to be there. And so getting lost is also a hazard.
Not an option.
Not an option, but a hazard, yeah. So I'd never encountered a bear. I kind of wanted to, but maybe like from far away, since maybe this year.
Well, yeah. We're talking about the kind of the tools of the trade. I hadn't really thought about a party straw as one of your instruments, but I thought maybe a shovel was one of your tools. What other types of equipment do you use in your work?
Right. So definitely our work goes from the shovel, which is maybe the low-tech end. I don't know, maybe the party straw is the low-tech end of the spectrum. Up to maybe the most high-tech is an accelerator mass spectrometer. One of our main tools to study carbon is to look at the carbon isotope. Because if you see CO2 or if you measure CO2 or there's carbon in and out, you really can't track an individual atom unless one of the ways we do it is by using isotopes. So if you have a C13 isotope of carbon or a C14 isotope, you can trace that. But C14 is present in parts per trillion. So it's really very scarce, and you have to use an accelerator mass spectrometer to measure it. So that's one of our main tools. We prepare pure graphite out of field samples, send them to an accelerator, get our C14 numbers. So
is some of that work done here?
So some of that work is done here, and the accelerator is at Lawrence Livermore National Lab. And so we collaborate really closely with them.
So this is carbon-14 dating, as like in
the way an archaeologist would do it.
An archaeologist would use.
Much the same.
And is that sort of time information turned out to be useful in your studies? Is it helpful in, say, tracking the levels of carbon that are sequestered or over a longer period of time? Are you finding anything?
Yeah, so the same way that an archaeologist can use radioactive decay to know how much how long a grain was in a vase or in a, in a container in a Greek site. We use C14 as a clock for how long ecosystem carbon was in the soil. So we use that radioactive decay as a clock. And one thing that we've learned in our work is how long soil organic matter stays. In most soils, if you get down below the surface, you have organic matter that's there for tens of thousands of years.
Tens of thousands?
Tens of thousands of years, unless you plow it. I mean, unless you do something, go in and disturb it.
No,
it's incredibly stable.
Was that an unusual finding? Was that what people expected?
Yeah. No, I think people expected it to turn over much more quickly than that. People had thought maybe hundreds of years. But we find samples that are almost depleted in C14, so that's 50,000 years old.
Wow.
Yeah.
And
it's really...
one of the things I read in some of the materials you sent me about your work that surprised me is that apparently most of the carbon cycle activity that takes place in soil takes place fairly near the surface. You can have 3 meters of soil, but it's really in the upper 10th or so. How does that work? Or maybe I've misread your research.
Well, soils are, it depends on the depth, depends on the rock, but let's say they're anywhere from this deep, 20, 30 centimeters to a few meters. But all the plant roots are mainly, the plant roots are living at the surface. That's where the nutrients are, that's where the rainfall first comes into the soil as moisture. So you have all the roots are concentrated up at the surface, and that's sort of the action. And most people just study the surface. And one thing we've done in our work is take the shovel and dig a little deeper and learn that it's actually kind of a different world as you go down, that there are different microbes, and that's where the organic matter is staying for, as I said, for 10,000 years, even though all the organic matter thermodynamically should decompose. Just like the food that we eat. I mean, plant products have a lot of energy in them.
Well, so if you have this very ancient organic matter just a few meters down, does that mean that it really isn't cycling, or is it just a very slow carbon cycle?
I think it's a very slow cycle. And that it just depends on the microbial community and the whole ecosystem there, what the climate is, the mineralogy, and that's controlling the rates. But I think it's dynamic. Yeah.
So let's go traveling again. You mentioned that you do work in the Great Plains. And there it may not necessarily be soils work, as you said. What are you looking for in the Great Plains?
Yeah. So in the Great Plains, our study is atmospheric carbons. We're studying a very large region. It's the size of an old GCM grid cell, which is about 300 kilometers by 300 kilometers. We have a big team project, and some people are here, Sebastian Barod and Mark Fisher, and several people helped us set up a coordinated suite of instruments throughout this very large region. We have an aircraft flying with instruments, we have a tall tower, and we're measuring atmospheric composition and the land surface, and trying to see how does the land surface really influence the atmosphere. Can you pick it up? Because I think for a lot of people it's a bit surprising that anthropogenic activities, our human activities, can change the atmosphere, the way that we're causing the rise in atmospheric CO2. So here we're looking specifically at what are these plants doing and what's happening in the atmosphere. And in fact, if you look at multiple species, you look at CO2 and CO, we look at C13 in the CO2, you see a distinct signature of land use, what we're doing there in the Great Plains, wheat fields, pasture, on the whole atmospheric profile. So you see the CO2 being drawn down in the summer.
So essentially right above that kind of activity you're measuring?
Yeah,
the
but all the way up to 15,000 feet. So that's the airplane part.
Okay.
Yeah. Yeah.
You mentioned not that far away the Colorado Rockies, another different environment, mountainous environment. Is the science different up in a mountain than it is down on the plains? Yeah,
as I was saying, we're sort of opportunistic. So we're looking at each site, what can we learn here? And what we're studying right now in the Rockies is a different question on the carbon cycle, which is what will climate change do to forests? And in particular, as it warms, can forests really march uphill? Will we get the subalpine forest marching in the sense of trees, seeds falling, trees growing, new forests emerging higher up in the alpine? And if you'll forgive me, one more prop.
Sure.
Thanks to the dining hall. This may not look like an ecosystem experiment, but this is a French fry. And if you're trying to think about how would you simulate global warming. How do you simulate and look at the effects of enhanced infrared radiation? So we have more greenhouse gases in the atmosphere, more downwelling IR. Well, we use IR lamps in the cafeteria and in many eating places to keep food warm. So if it's not fresh out of the fryer, it's sitting under an infrared lamp. So a professor who was a mentor of mine, John Hart, got the idea that we could get these lamps from a restaurant supply and suspend those over the soil, and then we would have an infrared heating experiment. And that was part of what I did for my dissertation, was to set that up. It's now running 20 years later. It's still going. And we have this new experiment now in the Rockies, planting seedlings. And this is a big team effort of many people. Warming the soil, the snow melts earlier, and the seedlings dry out. So even if the seedlings do move uphill, there are real constraints on whether or not we can get new forests. Whereas on the other end, the southern end of forests, it's getting very warm, and we're worried about contraction when we lose those forests.
We have a rule here of no PowerPoints, but
Yeah.
we do allow props, and I've been staring at this this device, which is
Right.
looks a little more high tech than a French fry. What exactly is this?
Yeah, this is not from the cafeteria, actually. This is an infrared gas analyzer. So any of you who know anything about climate change will know that it's being caused by increase in greenhouse gases. Greenhouse gas means absorbs infrared. So you can make a device with a laser, in this case an infrared laser, and that light is absorbed if there's CO2 in a cell. So this device is actually up in here is an infrared laser with a mirror. So it's a very long path length, but it's made for field work. So this is, again, a device for the field. And this part you can put around a plant leaf, capture it, and you'll actually measure the CO2 exchange with the atmosphere right there. So you're measuring the photosynthesis rate.
This is a living plant.
A living plant. Or you can take this same device, this upper part, and put it on a chamber that looks a bit like an old coffee can, put it over the soil and measure soil respiration. So you're getting a real-time measure of CO2 flux. And you can take it anywhere that you can carry this and the battery and the little computer that goes with it.
So what will that set me back when I go to Radio Shack to pick up one of those?
No, that's about $24,000.
$24,000?
Yeah.
Okay.
But, well worth it. Yeah.
Well, you know, we talk a lot about some of the journeys that you take in order to do your science. I was kind of intrigued in reading your biography of kind of your professional journey, that you didn't start out thinking you were going to be a scientist, and you worked in your family's store in San Rafael. Tell me a little bit about how you got to be here.
Yeah. So for 3 generations, my family had a food business with nuts and dried fruits, a farm, nuts, dried fruits. And then we had a food store, and I worked in the food store, and sort of the plan was I would keep working in the food store and inherit it from them and keep going. That was called the Torn Ranch, that had our name. And I just started spending too much time out of doors. Sorry, I think that's me. And just fell in love with the environment, said I have to do something that I feel is contributing to understanding the environment and just working in it. But I still had to work in the store. So as you say, then I went to community college and still kept working full time. So it took me a long time to get out of school.
But I think a lot of people think that the path to science is elite universities, and of course you did go to Cal. But I mean, did you find that College of Marin was a good experience for an ambitious young woman?
Yeah, maybe I didn't know I was then. But, yeah, it's a great. I mean, going to community college, you have small lectures of teachers who are there to teach you. They don't have another job of, say, doing research as well, and the demands. So the classes were small, and they were great. So, we've had a student. I just say that we've had several student interns in our lab who go to community colleges as well, and they've been great. So.
As a Marin County teenager and a person who is able to kind of pursue your passion about saving the earth in so many ways, did you ever find that conflicts with the sort of the scientific ethic that you simply have to observe what's out there and report the results? Do you ever find a conflict there, or do you find it actually fits in very well?
Yeah. That's an interesting question. I mean, we already talked about the flight miles. So,
Right.
and when you're doing an elevated CO2 experiment, let's say you want to know the effect of rising CO2 levels on a plant, you end up emitting an awful lot of CO2 to create your elevated CO2 experiment. But no, I think, I mean, for one thing, on a very practical level, the footprint of our experiments, yes, we go out and manipulate the environment, is really very small compared to what we need to do on the land to produce food, to graze animals. We produce fuel, fiber from natural ecosystems. So many systems are managed. And actually, our footprint is, I think, is pretty small. And you don't want to go totally change the ecosystem anyway, because then you're not learning what you set out to learn. So you're trying to have kind of a low impact as you go, except for the flights. Yeah. Now that you've mentioned that, I won't be able to forget.
I remember attending an exhibit at the California Academy of Science where they do the carbon footprints, and it was essentially a balance scale, and almost all these wonderful things you could do, you can improve your carbon footprint, but as soon as you hit the airlines, it just totally blew it out of the water. It's very discouraging.
Yes.
But what about the science of the carbon cycle? I mean, do you find it as sometimes just depressing to see what, the kinds of findings you're getting, and does it make you fear for the future? How do you balance that?
That's a good question, and I think I'm going to come back to that in sort of a roundabout way, that when people are thinking about what to do, the advice I've heard, and the advice I think is really good, is that you pursue what you love. You work on what draws you in aesthetically. And that's a saving grace, because then as you're finding out that there are really big risks, that ecosystems may generate positive feedback and amplify global warming that we're causing, or you find things that really are scary, at the same time you're working on what you love, and it's sort of, there's an aesthetic there that I think keeps you going and makes it very rewarding in that sense, and not depressing.
I noticed one other item in your in your collection of show and tell, and this appears to be a root. It's probably a little difficult for people in the second row to see this, but roots just must play a central role in your studies. And what is the function of a root in the carbon cycle? I mean, I think we think a lot about carbon cycles and leaves and photosynthesis, but what about roots? What role do they play, and are our friends the microbes involved in those as well?
Right. So this is a root from a coast redwood. It's a Sequoia sempervirens. If some of you are visiting California this summer, then you may not know it's the tallest tree, taller than the Statue of Liberty. It's the sort of standard old growth sequoia. And, but they produce little tiny roots, just like any other tree, and they're very, very shallow. It's just enormous tree, small little root system. But what we want to know is, how long do these roots live, and how long do tree roots live in general? We know about the wood, that's easy. But you can use C14 as a clock for the roots as well. And we found out that these little fine roots, if you can see them, they're about 1/2 millimeter in diameter, and they were thought to turn over on sort of months, grow and die. They can live 10, 15 years. So the tree or plant is really investing in these roots. From the point of view of the carbon cycle, it means that trees pump less carbon below ground than we thought.
Less carbon? Less
carbon below ground than we thought, but at the same time we're learning that this is the main source of soil carbon. So there must be this very efficient transfer from the root to the soil, because we're putting in less than we thought, but we still have a lot there. So it means we have very efficient retention. So it's sort of a simple story, but it's changing how we model and understand how trees grow and what might happen in the future. So put that back.
We also wanted to make sure that you folks in the audience who have any questions about the carbon cycle and about Margaret's work, please ask them. We have Dan and Julie with microphones. If anybody has a question, this would be a good time to ask.
You mentioned that you did some research in Russia, and you had some samples from 100 years ago that you could compare with. What sort of patterns did you notice between the 100-year sample and what you found in the soil? Well,
one thing that had happened in those 100 years was the area that we sampled had been preserved, and there were no more wildfires. It had become all wheat fields, and so there was no more burning and no more input of black carbon or biochar. And one thing we discovered was that in that 100 years, the soils had lost about 25% of the black carbon or biochar that had been in the soil. And that's one way that we know it does decompose from the soil. We found an accumulation of some trace metals that indicated some metals that had been used in weapons production. I think I was mentioning that in the region. And beginning to think that, it's a small thing, but that calcium and the changes in the calcium in the soil as precipitation change were responsible for a lot of changes in soil structure that we were finding. So that's something we're still looking up for the calcium. But otherwise, as a soil profile, the ones in preserved landscapes were just the same, you know, to the centimeter of thickness of each layer.
Another question over there.
You mentioned the biochar degrades much faster than people had claimed, but then you also said that if the carbon gets down a meter or two, then it stays for tens of thousands of years. I guess that leads to a question of what's the best way to pull carbon out of the air and put it down a couple meters so it'll stay there for tens of thousands of years.
So what you just mentioned is kind of the central conundrum in our work, that any plant material, black carbon, really anything you find is just thermodynamically unstable, favorable for decomposition, and yet this fundamental question, why does organic carbon persist in soil for 10,000 years? And what we're finding out, and I think pioneering here, is that it's an ecosystem property. It's not a property of this little molecule. You can't breed a plant to produce more lignin or char material and just put it in. It's an interaction of that material, the mineral matrix that it's in, the microbes that you have, the climate, and that's what gives you a decomposition rate. So it's much harder to predict, and it can be very, very slow. So then back to your question. I do think that carbon in deep soils is preserved very, very well, and that roots are probably the only ecologically sound way to get it there, because otherwise you're digging up the soil, which causes decomposition, and that's what you're trying to avoid. So the most simple answer would be deep roots and preserving the plants that you have that are growing now, because that's the established root network. That's the way to go. But I was just at a meeting in Venice with a group of people. This is for the IPCC Working Group 3, who are interested in climate mitigation, and they wanted to know about what's called carbon dioxide removal. It's almost a form of geoengineering, as they're saying it, which is that the CO2's out there. What can we do now? Now that we've let the cat out of the bag, I guess, how do you get it back in? And plants do take CO2 out of the atmosphere, but there is so many competing demands for ecosystem services that I don't know that's something that we'll start studying more, but it's not like there's a whole lot of landscape that's just waiting to be managed differently, say for our purpose of taking up carbon. And I don't think we should count on ecosystems as getting us out of the mess. Even though plants are very good at taking CO2 out of the atmosphere, fixing carbon requires water, it requires nutrients, it requires land, and there's just an awful lot of demand for all of those resources.
We had a question over on this side.
You mentioned methane and the permafrost.
Yeah.
A few years ago, there was a scare about in Russia about a huge potential for the huge release of methane from the permafrost. Would you comment on that?
Yeah, I mean, the greatest way to get the sense of that is there's a YouTube video of people out in the permafrost. It's a snow-covered area, and there's a thermokarst lake, a little shallow lake underneath, and somebody takes a match and lights it by a hole in the snow, and it just erupts because there was so much methane coming out of this thermokarst lake. You know, and everyone jumps back. And if you do field work in Siberia, I don't know if it's true, the tale is that you can heat your tea, your samovar, with your tea in it on a methane flame. I don't know if that's true. But you have what I do know is true is that you have an awful lot of organic carbon in the soil, that as permafrost thaws, that material is more bioavailable, and the conditions are very conducive to methane production. So the questions are, how fast can that permafrost degrade? And to what extent will that methane be consumed right there in the soil by other microbes, or find its way out through the party straw or the plant? And those are difficult questions about how the whole landscape will evolve. But starting in one month, we're going on a scouting trip in Barrow, Alaska, and then on the Seward Peninsula near Nome to scout for field sites where we'll be trying to look into that question in a 10-year experiment. So then maybe we'll know more. 10 years.
Opposite side over here.
Yeah.
You mentioned that the microbial community, the microbes in the soil, are not well understood, that there's a billion of them per gram or so, and that there are many different types, and that it's not possible to culture them, which surprised me when you said that. So I'd like to understand more about why it remains so mysterious.
Yeah. Well, the simplest reason about culturing is imagine the complexity of a soil ecosystem. You have fresh root inputs coming in, you have films on a mineral matrix. It's very sort of delicate system. The minute you bring it in the lab, that's gone. You have all this abundant oxygen, it's probably drier, maybe different predators or not. And so it turns out not just to be difficult to culture them, but to know you've done it. How do you know? Okay, you've got something growing. What is it? You can't go to a little plant. You know, for plants, you can go and you key out the flower or how many needles there are. And that didn't exist for bacteria. You know, where would that come from? Until you can get at the genes. So now with the Joint Genome Institute and some other institutions, you have the ability to look at the DNA and the RNA and say, what is this? Where did it come from? What other microbes is it related to? Is this an ancient phyla? You know, that kind of question. And so for more, though, I'd have to turn it over to Owen, who's really an expert in not only what's there, but how does it relate to the environment? How does environment control the behavior, then, of this amazingly complex mixture of microbes? So then it'll be your turn. I mean, at least to come, you'll have to come up here someday.
I think that's a great question. We've struggled with trying to characterize microbes from natural environments for that reason, that they're not culturable. I think the estimates go from somewhere from 0.01% to maybe 20% if you put in a lot of time and effort into culturing those organisms. But that's the number of organisms you can culture from soil. The diversity is far lower. So there's our whole branches in the phylogenetic tree that are not represented by any cultivated isolates. So people like Jonathan Eisen and folks at JGR are trying to get the genome sequences for these organisms. Not only does that tell you what type of organism it is, but it also gives you some information about how you may culture those organisms. So there's a feedback.
Yeah.
I just have sort of a follow-up question of that myself. Yeah. If you are able to use genomics to identify greater numbers of these microbes in these microbial communities, can you also get a little sense of their function through the genetics?
Yes.
I mean, it's one thing to be able to count the bugs.
Yes.
But what about actually being able to know what they do? Are we making any progress in that field to sort of further understand the ecology, the microbial ecology of soils?
I think you'd have to say probably in the last 5 years that has just taken off, this challenge and being able to meet it of not just what's there, but what function do they play in the environment. And you can do that initially just in the lab. What proteins do they produce? What enzymes are produced? And now you can go out and get field samples and do what's called proteomics, and all the proteins that are produced. And it's a nice melding of very basic science, say in the genomics, with very applied science. The JBAY, the Joint Bioenergy Institute, is using that kind of information to say, can we use microbes to help us convert plant material to cellulosic biofuels, just as an example. But really, they're developing a lot of techniques for understanding the function of microbes.
So there could be some very practical spin-off from this field research.
Right. Yeah.
Question over here.
Yeah, going back to carbon sequestration, I was wondering, you said that once the carbon's down in the soil, whether or not it remains inert is a sort of a function of ecosystem properties. If we've already profiled the soil and know about the ecosystem properties, how safe of a bet is it that they're not going to change, and that you know, once we get the carbon down there, that it'll stay down there?
That's a great question. So once either you find old soil organic matter deep in soil, or you can put it in there where you think in today's conditions it ought to be stable, what will happen to it? And we have a real challenge that in the next 100 years, the predictions are that Earth will be quite a bit warmer, and that rainfall regimes will change, so soils will be wetter or drier than they are now. And we really don't know how that will affect microbial activity and sort of accessibility of carbon to microbes. So we're trying hard to learn how to model that and how to predict it. But it turns out to be a very difficult question, because if you go to natural environments, they're in a sort of semi-equilibrium that's established over thousands of years. If you do a little incubation in the lab, it's done over 1 year. And we want to understand this kind of evolution over decades. So I think that vulnerability question is really open. You should do research on that.
Hi, can you talk a little bit more about the influence of the mineralogy on the decomposition rates?
Yeah, I got Becky Sutton sitting next to you, put you up to that. She's hiding. So it turns out to be rather complex, that compounds that you think would be very labile, like a protein or a sugar, if you put them in a petri dish with a microbe, they will disappear in hours. But instead, you put that same material into soil, especially soils that I've worked in, like in Hawaii, that are volcanic ash, lots of surface area, lots of reactive aluminum, and they form the carbon forms very strong bonds, or interactions on that mineral surface, and the microbes don't seem to be able to then decompose it. It's a very small, so-called labile material. Don't ever believe that now. If people are telling you something's labile or recalcitrant, just throw out the terms. It depends on what's happening, and these mineral surfaces seem to be able to stabilize the carbon for a very long time. And I think vintners know that. I think that there's fields in which people use different kinds of minerals to bind proteins and get them out of soil, but we're just learning how important that is, or I should say, get them out of wine. But we're just learning how important that is in the soil environment. And that's one reason that I worked in Hawaii, because in Hawaii you have these, the islands are all different ages, and they're all formed by this plate moving over the same tectonic hotspots. You have the exact same parent material coming up, being formed, but you can look at ages. We looked at sites that were 300 years old and sites that were 4 million years old.
So you have a natural laboratory.
A natural laboratory.
The age of the islands as they pass over that hotspot, very interesting.
I mean,
what have you found are the oldest? The soils in the oldest islands the most carbon rich, or what?
No, so it peaks in the middle. So you get this weathering, and you get very much more reactive minerals, a lot of iron and aluminum on the surface. They can bind the carbon or have some interaction with it. And then as the soil gets old, it weathers, it loses the charge, and actually the ecosystem loses carbon. So you'd have one site with half as much soil carbon as another, even though the plants were just as productive, and it was the mineralogy that had changed and could no longer sort of sequester or protect the carbon.
So going back to the original question, mineralogy plays a huge role in carbon sequestration.
Yeah.
And in the ecology of the soils. I'm just kind of curious about how do you define soils? It sounds basic, everybody knows what dirt is, but I mean, what about the soils next to a swamp? Does that count because it's halfway underwater? Or what about the soils at the beach? Do they qualify? Does a beach qualify as soils?
There are several people here who know much better exactly how to define a soil, like Uma Khan. But I think for this purpose, the soil has been, it's not the bare rock. It has been changed by biological and chemical and physical activity to become something different. So it's, again, a biogeochemical process of some kind of weathering. And you have organic acids from plants that are causing changes. And you begin developing some kind of structure that reflects that environment. So you grade then from soils to sediments. You were getting into sediments, and I don't do sediments. I'll leave that.
Well, yeah. when I think of soil science when I was in high school, it was almost all agriculturally driven. Soil was all about topsoil and the fact that we were losing topsoils. Well, let's go to that basic question about our topsoils. Is there something about topsoil? We know it's great for farming. Yeah. What about for the carbon cycle? Is the fact that it's great for farming also make topsoil great for the carbon cycle, and hence should we be worried about the carbon cycle when we're also talking about the worldwide loss of topsoil, for instance?
I mean, now I'm not going to remember the quote, but didn't Franklin Delano Roosevelt say something about the country that takes care of its soil? Oh, I'm really going to get that wrong. Takes care of.
Not at my fingertips.
Okay, someone probably knows that out there. But the topsoil is where the fertility is. That's the nutrients. That's the water holding capacity. So that's where you need to grow crops, and it's the part of the soil that's vulnerable to disturbance. If you plow or remove vegetation, you have erosion, and you're losing that topsoil. And it's very, very slow to regenerate. So it's not that it's completely a non-renewable resource, but we really ought to treat it as a non-renewable resource, that we really protect the topsoil.
But is the best topsoil also the best carbon recycler or cycler?
Oh, that's a good. Well, to the extent that when you say the best topsoil, that's probably meaning the most fertile. So you have the most productivity. So you have the plants are taking CO2 out of the atmosphere at the highest rate. I think that's usually what we mean by good topsoil, because we care about plants again, because we like to eat, have all the goodies. So yeah.
Fishing for questions out there.
There's natural absorption of CO2 in the biosphere, and I was wondering if you could, what fraction of that is terrestrial versus oceanic? And I'm wondering, related to that, is there a terrestrial analog to ecosystem impacts like ocean acidification?
Got it. Yeah, good question. So I think probably the first part of your question is referring to the fact that right now so we're releasing humans release something on the order of 9 petagrams, or 9 times 10 to the 15th grams of carbon every year as CO2 to the atmosphere. But only half of that is staying in the atmosphere. About half of it each year is taken up by the biosphere, by the ocean and terrestrial system. So we're getting this huge, we're being saved from ourselves, this huge benefit by having natural systems taking up about half of the carbon that we emit. It's often called a carbon sink. And just about 50-50, half of that's going in the ocean, and half of that is going into the land. And it's a really important point that when people think about the effects of anthropogenic emissions, of our CO2 emissions, they are assuming that sink continues. But the fact is that we really don't know where the carbon is going, and we don't know why. So it is then clearly, we can't predict how long will this happen. And as John Hart says, we don't know when the sink will clog. And then what happens is we bear the full brunt of all of our emissions. So then I think the second part of your question was referring to that if you have more CO2 dissolving into the ocean, you get more acidic water. And that makes it hard for many different kinds of organisms that make their skeleton or exoskeleton or shells out of a carbonate. It makes it harder to precipitate that material, harder for coral reefs. So on land, you might say, what's the problem with more carbon? And to some extent, if you're thinking about soils, there isn't, at least from an anthropogenic point of view. Generally, more soil carbon is more fertile, but it's how you get there. If you try to increase carbon storage in a management sense, you're probably adding nutrients or water, and then you're changing a lot of things and making the system more vulnerable to disturbance. But I think there is a limit to how much carbon ecosystems can absorb, much more than for the ocean. And we don't know what that limit is, but it probably has to do with light limitation. You get a closed canopy, you just can't fix more. Nutrient limitation, you've already taken up the available nitrogen. And probably the current sink is a response to past disturbance. We've deforested, like we deforested the Northeast. There's disturbance and deforestation in the tropics that are regrowing, and that's probably taking up the carbon. So it's quite likely that what we call this terrestrial sink is not sustainable, as sort of just a natural fertilization effect. And it's because it's a carbon cycle. The more carbon plants fix, that material is shed into the soil and microbes decompose it and you get CO2 release. So that cycle means that eventually those systems will probably stop taking up more CO2, and it's not the savior we should count on, in terms of taking up half of our emissions every year.
We have perhaps time for one more question, if anybody has one out there. And if not, I'm just wondering about, we often talk about carbon cycles as kind of a stable notion. A cycle is something that's stable. Yeah. And when we're talking about climate change these days, we're talking about instability. I'm just wondering, is it possible that what we think of it right now as a stable carbon cycle could be sort of revved up at a different level, right now we have an instability, but could there be another plateau, a very different sort of carbon cycle with different degrees? I mean, I'm thinking perhaps of another planet even. Is it possible that maybe historically there was a stable carbon cycle that looked very different from what we have now, I think is what I was trying to say.
Yeah, indeed. Indeed, we can think about the era of the dinosaurs, the Cretaceous, and think about times in Earth's history when it was warmer and wetter and we had much more CO2 and methane in the atmosphere. I mean, Earth has gone through very cold glacial cycles as well, but we've been through much warmer cycles. Now, if you think about just the last million years, you think about human history, we're now at just about as warm as it gets, and certainly more CO2. But if you go further back, go 60 million years back, then you'll see that, yeah, we can have a Earth that goes on to produce new species and, you know, have a future that lands us here with much higher CO2. But it's important when you look at those past worlds or past Earths that they do look very different. And so if we want to count on humans being supported by that Earth, then we have to think of, you know, does that work for us? But definitely.
Well, before we wrap things up, I want to point out that we're going to have Marion Fuller, who is Principal Research Associate at EETD, talking a different aspect of energy, talking about efficiency for sale, who's buying, kind of the science not only of trying to explain how people can save energy, but actually getting them to buy into the process. I think it'll be a fascinating discussion, and that won't be for 3 weeks. It's on July 27th at this same place, at the same hour. And so, first of all, I want to thank you, Margaret, for coming. It's been a fascinating hour, and really appreciate your time here.
Thank you. Thank you.
And I also want to thank the audience for coming. Some of you have been here to all three of these events. If you enjoyed it, tell your friends.